coach. Yeah. All right. Hello, everybody. First of all, thank you all for coming this afternoon. It's probably the first sunny day in a couple days, so we're kind of hopefully on the backside of all the cold weather. But I'd like to introduce to you all Dr. Jocelyn Colella. Jocelyn is on the faculty at the University of Kansas Department of Ecology and Evolutionary Biology and has an appointment also in the Department of Museum Studies. She is also the curator of mammals at the University of Kansas Biodiversity Institute and the Natural History Museum. And she's also a research affiliate with the University of New Mexico Museum of Southwestern Biology. Jocelyn describes herself as a collections-based evolutionary biologist that synthesizes data from genomics, morphometrics, and ecology, using all aspects of the extended specimen to answer her research questions. Her specific areas of research interest include the interplay between diversity, speciation, and selection, adaptation to extreme environments, repeatability of evolutionary outcomes, and linked evolutionary dynamics among host and parasites. So she has a good website. You can copy and paste all that information to your introduction next time. Her research has been published in a lot of different journals, including the Biological Journal of Linnaeus Society, Bioscience, Heredity, Journal of Mammalogy, among others. I've been following Jocelyn's career now for a little over a decade. She was starting her graduate career at the University of Mexico as I was finishing mine at Texas Tech, which definitely ages me a bit. If you know anything about those two institutions and their history in mammalogy, it is highly interwoven. In fact, I think we were academic first cousins, one time removed. Fortunately, we've become friends and colleagues and have been able to publish a couple papers together on the Museums and Emerging Pathogens in Americas Network, which I'm assuming she'll probably talk about a little bit. So please join me in welcoming Jocelyn to Michigan.
Thank you, and thank you for the invitation, Cody. Can you all hear me? And you can, not so well. Let me see what I can do. There is no crank it up. Can I crank it up here? Mic volume, look at that. Can you hear me better now? Thank you. Oh. That's Cody, not me.
Better now.
Okay. Thank you.
Is that good?
Or is there too much feedback?
Yeah.
Go one. Better? Better. Okay, thank you. I appreciate that feedback in the beginning so you can actually hear my talk. I have so totally enjoyed my visit with all of you today. I've had the chance to meet with a number of you in my jam-packed schedule, thank you, Cody, where I ran between every single hall of this building, and I had an awesome lunch with all of the grad students. So thank you for making it to my talk this afternoon. As Cody mentioned, I'm at the University of Kansas, which can be confusing because we abbreviate it as KU, even though it's University of Kansas. And I run a collections-based research program where I use mammals as models to investigate change through time. And I do look at mammals as well as parasites and pathogens. Cody mentioned that I lead the Museums and Emerging Pathogens in the Americas Network, along with Cody. And I'm not going to talk a lot about that group today, but I will highlight it in my RMC or RMT talk tomorrow. RMT talk at the RMC? Unclear. Sure. So if you want to know more about MEPA, I will talk about that a little more tomorrow. So to get started, I want to acknowledge that the work in my lab is highly collaborative. It would not be possible without this amazing team. I have about 20 people that I work with regularly, including full-time staff members, 2 postdocs, 7 graduate students, 2 postbacks, and an entire cohort of undergraduate researchers. And I really couldn't share what I'm about to share with you today without the support of all of these amazing humans. I will have an opportunity to highlight some of their work throughout the talk, and I will do that. I'll give student shout outs, but I just want to acknowledge them first because they do a lot of work. To get started in some of the research, you may have heard this quote, or at least a version of this quote before, that the only constant in life, and in evolution, I'll add, is change. And this quote acknowledges that the universe is in this constant state of flux, and everything in it is part of this constant process of change. And although this was proclaimed by a Greek philosopher, it resonates with me as an evolutionary biologist because one of the most simple definitions of evolution is change through time, right? But that change can occur across many different dimensions. It can occur across time and space and taxonomy, across genotypes, phenotypes, phenology, and physiology, and so on and so forth, as I'm sure many of you study. Museum research collections, like the one here at UMMZ, and also the one that I curate at the University of Kansas, capture many dimensions of change, biological and evolutionary change, by sampling species repeatedly across space, time, and taxonomy. And these collections are especially powerful for assessing change when they're built holistically. So that is when the person sampling wildlife, for whatever reason, intentionally maximizes the scientific value of each sampling event by saving all parts of an organism, not just the one part that they need to answer their research question in that moment. For example, at KU, we save traditional skin and skeletal material of mammals. We also do fungal swabs and preserve multiple different types of tissue, including lung, liver, kidney, spleen, heart, other muscle. And we do this in multiple different media, including RNA later, or DNA RNA shield, or even preserved cryogenically in liquid nitrogen in the field. And we do this both domestically and internationally. Plus, we have ecological field notes, standard physical measurements, internal, external parasites, among other avenues of data. And I had some meetings today where I talked about adding possible blood or serum treated in certain ways for different treatments. That is the purpose of museum biorepositories. These materials can then be extended in many ways, using genomics, but also morphometrics, isotopes, ecological modeling, to name a few. But notice that the way in which a specimen or a sample is collected 100% determines how it can be extended. If we only collect one piece, we can only extend that piece so far. So finally, all of those derivative data generated by researchers, things like genomes, phenomes, CT scans, isotomes, what have you, the types of data that we generate can then be tied back to these physical and digital specimen records, not only to answer the initial question for which that organism was sampled, but also to facilitate cross-disciplinary synthetic observations as more questions are asked by more and different researchers, and that data comes back to that single specimen record. So we can make bigger observations than each of our independent science questions in isolation. My lab leverages the global network of mammalian biorepositories to ask, is evolution repeatable? At least, is it repeatable for mammals and their symbionts? So that is, do organisms respond to change in consistent ways? Are there common factors, for example, in host pathogen or host parasite relationships that can predict spillover or cross-species transmission? Do species with similar life histories respond to environmental change in similar ways? And do we see the same evolutionary outcomes occurring across different hybrid zones? The same thing is happening. Do we see the same evolutionary outcome? And our results inform both the fundamental study of evolution, so basic research, as well as applied wildlife management, conservation, functional genomics, and One Health applications. So today, I'm going to touch on 3 dimensions of change that are under active investigation in my lab. That is change over taxonomy, change over geographic space, and change over time. Which these 3 items will scaffold my talk today with the 3 dots, oh no, you can hardly see them, in the left-hand corner I'm going to use to indicate examples that we're on. I may shift them up. So first, from a taxonomy dimension, I'll give you a little bit of a teaser of some of my viral discovery work in mammals. Then for geography, we'll look at changing risk landscapes for hantaviruses in Panama. And last, to investigate change across time, we'll examine replicate hybrid zones among North American red-backed voles in the genus Clethrionomys, or perhaps you've heard the genus Myodes. Obviously, I had a hard time choosing what to present to you today because I do a lot of different things, but I figured with the pathogen biorepository here and the Natural History Museum, I wanted to at least touch on both some of my pathogen work while also touching on some of my classic evolutionary genomics work. So I will start with our taxonomy example. So a symbiont, hopefully you're mostly biologists, maybe. You may be familiar with this term. This is just an organism that has a close association with another organism. And in the context of my research program, that other organism is the mammal host. I work on mammals, pretty much always a mammal host. Symbionts can be large, like fleas, ticks, mites, helminth worms, but they can also be microscopic, as is the case for many fungi, bacteria, and viruses. And a subset of symbionts that cause symptoms of disease in a host are considered pathogenic. So just because you're a symbiont doesn't make you pathogenic. A subset are pathogenic. And more than 75% of emerging diseases in humans come from what are called zoonotic pathogens. So those are pathogens that are transmitted to humans from non-human animals. And the vast majority of those come from mammals. But biomedical research has disproportionately focused on only a subset of all mammals. I'll call them the usual suspects, bats and rats, if you will. And although these groups definitely harbor many notorious zoonotic diseases, including rabies, Nipah, Hendra, and plague, they also represent the vast majority of all mammals. And as a result, there are two leading hypotheses to why we see more zoonoses emerging from bats and rats than we do these other groups. The first hypothesis is called the special reservoir hypothesis, which posits that there are a suite of shared ecological or evolutionary traits like body size, diet, or dispersal ability that make these groups especially effective reservoirs. Note that there is some controversy here surrounding the term reservoir. I did not name this hypothesis, so I will in this talk use reservoir and host interchangeably. Sorry. As an example, in support of the special reservoir hypothesis, flight is a trait that is unique to bats, at least within mammals. Sorry, birds. Flight is, as we know, energetically expensive, and it produces a massive amount of heat. Therefore, every night when bats leave their roost to go forage, their core body temperature raises to over 40 degrees Celsius or over 104 degrees Fahrenheit. This makes many bat viruses resistant to human fevers. So while there are clearly some traits, like flight, that may make bats exceptional reservoirs, there are also just a lot of them. Bats represent 22% of all mammalian diversity, and rodents represent another 40%. Therefore, if zoonotic pathogens are distributed evenly across all mammals, we would expect to see more or at least a proportional number in these species-rich groups. This is the idea behind the reservoir richness hypothesis. It essentially says there's nothing special about these groups, there's just more of them. So I am now leading an NSF-funded pursuit award that is testing these two hypotheses by systematically sampling across the mammal tree of life. To do this, my collaborators and I have run a greedy maximum mean discrepancy algorithm to select 1,000 branches based on minimum pairwise distances. So that is to optimize the phylogenetic distance. And then we're using geographic distances between species estimated centroids to break ties and produce a ranked list of taxa to sample. Those And taxa that we've sampled look kind of like this. They're spread evenly across geography, shown here on the bottom, taxonomy on the top. And this scale of sampling is really only possible through the global network of museum biorepositories, which have accumulated tens of thousands of cryogenically preserved tissues through both their own fieldwork and contributions from the broader scientific community over a long period of time. To date, we've borrowed more than 400 mammal tissues for this project. The first phase looks at 600, and the second phase looks at just as many. We performed total nucleic acid extraction, cDNA synthesis, library preparation with a viral enrichment step, and we've sequenced on a MySeq at the Goldberg lab at the University of Wisconsin, so they're in the veterinary medicine program. The reads were then assembled into contigs and translated across all reading frames. We then mapped reads to 2 virus data sets, 1 prokaryote data set and 1 eukaryote data set. Contigs that hit to either the prokaryotic or eukaryotic references but did not hit to the virus were thrown out as presumed contamination. And remaining contigs were then compared to 4 more databases. This includes 2 virus to microbe or VMR databases, 1 MCBI and 1 RDRP database. So RDRPs are the only universally conserved protein across all RNA viruses, as they are responsible for viral replication. This level of bioinformatic viral discovery was led by Dr. Yuri Wolf at NCBI. And now I'll give you a summary of what we've found so far. So of the mammals tested, at least 98 had at least 1 virus. Individuals with at least 1 virus, however, often had more than 1 virus, with a median of 6 viruses recovered per infected host. There were 4 notable outliers. The Gobi big brown bat, which is native to Central Asia, had an astonishing 132 recovered viruses. Ouch. Plus 3 rodent species outliers, 2 in North America and 1 endemic to Mongolia and northern China. In total, we've recovered more than 1,300 viruses across 45 different viral families. And of those, at least 24 appear to be novel, completely undescribed viruses. I won't say species. Viral taxonomy is very complicated. So, which is it? Is it this special reservoir or this species richness hypothesis that better explains patterns of viral diversity in mammals? Well, we're still in the process of assembling our trait data set, including things like body size, diet, longevity, among other traits. So we can't yet test the special reservoir hypothesis, but we can preliminarily test the species richness hypothesis. Of our 1,300 viruses, more than 800 were recovered from rodent species, and more than 200 were recovered from bats. That's way more than any other order. 1,300 viruses, 800, 200, you can do math. But we currently also have very different sample sizes from these groups. Instead of comparing those absolute numbers, we can use rarefaction curves to compare the relative viral richness from a set of sampled hosts. So these curves generally increase rapidly and then kind of plateau as only the rarest viruses remain to be found. So we're going to look at a few curves for a few different mammalian orders. Our expectation is that bats and rats will have higher viral diversity or higher species richness than other groups. Starting with bats, this curve has that characteristic rise in the beginning and is kind of just starting to plateau. So that might be what we expect. And remember, we expect these to have lots compared to other groups. So if we add Didelphimorphia, this is the order of opossums, we have fewer samples, which is why the line is shorter, but they are showing a similar pattern and perhaps plateauing even earlier than bats. So bats have greater viral species richness than opossums. If we add rodents, ooh, now it looks a little different. So we found almost 4 times the number of viruses in rodents than we have in bats, but we've also sampled more than double the number of rodents than bats. That said, it does have a higher slope and it may be plateauing, but seems to be still increasing. And last, if we add shrews, so this is the order Eulipotifla, we see something really interesting. The shrew curve is still increasing and even potentially surpassing that of rodents. There are only around 600 total described species within the order Eulipotifla, which is less than a quarter the total number of rodents. So from here, we can calculate an extrapolation of these curves to see if the pattern holds. And indeed, our preliminary results suggest that species richness alone does not perfectly explain patterns of viral diversity in mammals. And clearly, we need to look at more shrews during the second phase of this project. So while this is exciting, what we've really discovered throughout the sampling phase of this project is just how poorly we've sampled mammals across the globe. Or at least, if we're sampling mammals, we've done a pretty bad job archiving those samples responsibly. On the left here are the 1,000 taxa, mammalian taxa. I know you can't read it, maybe squint. The 1,000 taxa I wanted to sample in both phylogenetic and geographic space. These were generated by some of my graduate students, Danny and Alex, who both completed masters in my labs and now have continued into the PhD program. But here on the right is the reality of what we've actually found high-quality tissue resources for. And there are major phylogenetic gaps in primates and artiodactyls. So this is kind of ironic because primates are major biomedical models. They are used all over the world to test all sorts of evolutionary hypotheses, but also for drug trials and other animal testing. Also, artiodactyls are both livestock and game animals, which are regularly sampled by humans, but apparently not making it into public biorepositories. Geographically, we see gaps in Africa and Southeast Asia, but this may not be surprising when we think about the fact that these are some of the most biodiverse regions on Earth. So there's a lot to be sampled. So in summary, for this first third, we've screened more than 250 mammals and we've found more than 1,300 viruses, including 24 new viral species, taxa, or perhaps distinct evolutionary units. Our preliminary results suggest that species richness alone may not explain patterns of viral diversity in mammals, and we plan to expand sampling, especially for shrews, in the second phase of this project. But what this really means is that from a taxonomic perspective, when we're assessing change, particularly in host pathogen space, we need to look beyond the usual suspects, or we're going to be only examining a small fraction of the total picture. So by sampling just bats and rats, we're really biasing our own results. And further, this work importantly connects viruses to voucher specimens, such that as host taxonomy changes, viral sequences can be associated with updated names and updated distributions. This kind of permanent linkage of information is essential for public health action and informed risk management. Okay, so I'm going to shift gears now to assess change across spatial scales, geographic scales. And for this, we'll look at a case study of hantaviruses in Panama. Hantaviruses are a group of highly lethal zoonotic RNA viruses that are transmitted to humans through the fluids of infected mammals, mostly through aerosolized urine, feces, and saliva. New World hantaviruses, or those that are found in the Americas, cause hantavirus pulmonary syndrome, whereas Old World hantaviruses cause hemorrhagic fever with renal syndrome. You don't really want either of them. As far as we know, hantaviruses appear to be endemic to mammals. They're only found in mammals. They've been detected in rodents, shrews, moles, and bats. And each strain appears to be fairly species specific, so a unique strain in a unique mammal. Despite their presence in multiple mammalian orders, spillover has only been documented from rodents, with a 1993 outbreak in the US Southwest emerging from Paramyscus deer mice, which I understand we have a lot of in the collection here. And another in Panama in 1999, stemming from Oligoryzomys or pygmy rice rats. In these figures, Dr. Blas Armian and colleagues have visualized human case counts of hantaviruses in Panama across both space, geographic space on the bottom, and time, shown here on the top. The shaded pink region in the map on the bottom indicates the endemic zone of the virus, with most human cases occurring on the Azuero Peninsula, so on that south peninsula in Panama. And the Panama outbreak actually spurred the development of a collaborative wildlife surveillance program by the Gorgas Institute of Health in Panama. They partnered with the Museum of Southwestern Biology at the University of New Mexico, where I went to grad school. That same museum, Museum of Southwestern Biology, worked with the US Centers for Disease Control to respond to the US outbreak in 1993. And these partnerships have recently expanded to include me at KU, as well as Stephen Greiman at Georgia Southern University. Together, these field crews have sampled more than 13,000 rodents over the last 2 decades. Each rodent was serologically screened for the presence of hantavirus antibodies and then deposited holistically in a public repository or museum, with the goal of building a foundation of both physical samples and digital data from which we can better understand patterns of disease emergence. My team is now using these data to test how host ecology and environmental factors shape risk landscapes for people. And we're using hantavirus specifically because emergence is tentatively linked to climatic variation, with outbreaks occurring roughly 6 months after a heavy rain season. So increased precipitation is hypothesized to increase food availability, specifically for granivorous rodents, and that increases population densities of the host, leading to a higher incidence of spillover and disease, and increased frequency of human and mammal contact, leading to more spillover. We've now paired each of these 13,000 host occurrence records in Panama with environmental conditions that were present at the specific time and location of sampling to build suitability models for the host. The positive and negative serological information then allows us to extend those models to the pathogen, to the virus, using true presence and absence data, which tends to be a major issue when modeling hosts. You don't have as much negative data. This work is being led by Dr. Marlon Cobos, who's a postdoc in my lab, with support from PhD students Danny and Alex again. The Costa Rican pygmy rice rat, or Oligoryzomys costarricensis, shown here, is the wild host of Choclo hantavirus, which is the only hantavirus strain in Panama that has proven to be lethal to humans. So there are other strains that do not cause pathogenesis in humans. The shaded blue background in this map shows the geographic area that we have inferred as being accessible to the host. We then harvested, filtered, and thinned host and background specimen records, shown as triangles and circles here respectively, where background records are actually just other rodent species that can be trapped in the same type of trap. So they're usually similar in size, and that gives us a measure of sampling effort so that we can differentially weight non-detections of the host to account for sampling effort. Our serological results then indicate whether the animal was infected or recently or currently, and positives are shown here in red on the bottom. They cluster around the Azuero Peninsula again, which is where that 1999 outbreak occurred. We then use these time and spatially explicit data to infer suitability landscapes monthly from the year 2000 through 2018. Those are shown here for the host in blue and for the virus in red. These data are averaged across 4 months of the year, so 2 during the dry season and 2 during the wet season. And while there is not a particularly strong pattern associated with seasonality, there is a consistent drop in suitability following the dry season. And we see that reduction in suitability for both the host and the virus, so neither prefer dry conditions. But this is a 20-year data set, which allows us to examine responses to larger scale climate cycles, not just seasons. Specifically, El Nino Southern Oscillation Events, or ENSO cycles, which are global climate phenomena that are driven by variation in wind and sea temperatures in the Pacific Ocean. In the Americas, this generally leads to hot and dry El Nino years and cooler, wetter La Nina years. But in Panama, La Nina is experienced as mostly dry on the Azuero Peninsula, and this is due to a rain shadow caused by mountains on the western side of the country. Each ENSO cycle lasts about a year, and they occur every 2 to 7 years with varying intensity, separated by neutral, or as I would call them, non-ENSO years. So here are more maps of our host and pathogen suitability in blue and red again. But this time, I'm representing this for a strong El Nino year on the left, a La Nina year in the middle, and a non-ENSO year on the far right. Suitability landscapes for El Nino and non-ENSO years are pretty similar for both the host and pathogen. But host suitability shifts westward during La Nina cycles, which are shown here in the middle. La Nina also has a clear negative effect on the pathogen, which is summarized here on the right, which restricts its suitable area to just a small region on the Azuero Peninsula. Note, however, that there's kind of a curious disparity or mismatch between the areas of high suitability for the pathogen and those with high suitability for the host. This is perhaps most obvious on the Azuero Peninsula during La Nina climate cycles. This is in part because our red maps are not showing where the pathogen is or where it is not, but simply the environments that would be suitable for the persistence of the pathogen in the environment. In reality, the pathogen needs the host in order to be anywhere. So next, we wanted to get at where and how those host-pathogen interactions may be happening. And to do this, Marlon and I have partnered with Fola Augusto at KU and Helen Wearing at the University of New Mexico to combine traditional epidemiological modeling with suitability landscapes to build dynamic simulations. This will allow us to explore more realistic scenarios of hantavirus infection in wild host populations across the country. And to do this, we started with just a simple SIR model. You might be familiar from COVID or all of the MPOPI talks you've had recently. S stands for susceptible, infected is the I, and recovered is your R. So each of these are population states, and we can parameterize it with basic demographic parameters. We then included host and pathogen suitability landscapes estimated per month. Oh no. We're back. Estimated per month to inform parameters within our model, so including things like birth and infection rates. This allows those parameters to change over time as our simulation runs or as time progresses. For example, birth rate is directly related to suitability for the host, and infection rate increases when suitability for the pathogen is high. But this is just one population, and we have an entire landscape of suitability that varies across the country. Therefore, we used a meta-population model where each raster cell across the country represents a different population. For each population, we ran that independent SIR model with the addition of dispersal among cells and considerations of carrying capacity. We then ran this for the same time period that we had suitability landscapes for, so from 2000 to 2018. And I'm going to show you our initial results, again with 3 time periods, one in El Nino, the middle in La Nina, and then non-ENSO. But instead of suitability, here we're looking at the number of host individuals on the landscape. The effect of ENSO cycles here, again, is not dramatic, indicating relatively stable populations through time. These are rodents. They're pretty resilient. They can always find food, and they keep reproducing. But if we look at the number of infected hosts on the landscape, shown here in pink, we see many more infected hosts during El Nino and non-ENSO years. This visualization of prevalence of the virus in its wild host more directly translates into risk landscapes for people. So to bring this whole thing together, recall this map of hantavirus cases in Panama. I didn't make it, sorry, red-green colorblind. Where the pattern of human infection here is really not very aligned with areas that we found to be suitable for the pathogen. This makes sense in the context because we're not actually modeling where the virus is, just the conditions that allow it to persist in the environment. For it to be anywhere, it requires a host. But host suitability doesn't perfectly align with human case counts either. And this is where our simulations play a really important role in connecting models to actual risk of exposure. Because in pink, we can actually see where infected hosts are likely to occur on the landscape, which you might notice, comparing to the human case counts, much better reflects the actual geographic distribution of human case counts. So in summary for this section, we've demonstrated that hantavirus risk landscapes are not stable across space or time. They change dynamically in response to host and environmental cycling, with La Nina years significantly reducing pathogen suitability. We've also demonstrated that accounting for host, pathogen, and environmental interactions, as is exemplified under the umbrella of One Health, more accurately reflects risk landscapes for humans. Okay, so that's our coverage of taxonomy. Now we've covered space. Next, we're going to shift to change through time. So this is the last vignette. I'm shifting gears one more time. We've kind of seen time in all of these. It's a common theme. But this time I'm going to look a little bit more at deeper time, evolutionary time. We know that the Earth has experienced massive climatic cycling or change beyond just seasonality or even ENSO cycles, which I just talked about. For example, the Pleistocene epoch, which occurred 2.5 to 11,700 years ago, witnessed more than 20 hot, cold, or interglacial glacial cycles, as is illustrated by this oversimplified GIF. And the expansion and contraction of glaciers also shapes the demography and distributions of terrestrial mammals. For example, when ice sheets expand, populations or species may be separated by ice, leading to divergence through both neutral and selective processes. But when those ice sheets melt as the climate warms again, those previously isolated groups have an opportunity to come back together, and hybridization can occur as well as an exchange of parasites and pathogens. My lab uses genomic methods to determine whether we observe the same evolutionary outcome when those groups come back together each time. For example, there are multiple potential evolutionary outcomes of hybridization or genetic mixing. Hybridization can lead to reinforcement or the expedited divergence of the two hybridizing groups. It can also lead to homogenization or fusion of those groups. You can even see speciation of hybrid offspring distinct from either parental, although that's far more common in plants than it is in animals for molecular reasons, or even the formation of a stable hybrid zone with ongoing gene flow. Most recently, we've been assessing outcomes of hybridization between two species of red-backed vole, and this work is being led by the senior PhD student in my lab, Ben Wiens. Ben has recently accepted a postdoc at the University of Wyoming. And these two species are Clethrionomys rutilis, the northern red-backed vole, and Clethrionomys gapperi, the southern red-backed vole. They diverged about 2 million years ago and form multiple hybrid zones across the high latitudes of North America. So here are the geographic ranges of those two species, rutilis to the north in a tan gold and gapperi to the south in teal. And we can see at least these 3 big red dots indicate where we have sampled this hybrid zone in Southeast Alaska, Central British Columbia, and Southern Northwest Territories, Canada, to see if we're seeing the same evolutionary outcome in each zone. To start, we generated RADseq data because mammal genomes are pretty big, so this is a great place to start. We did this for about 100 voles that were loaned from 3 different museum biorepositories. And next, I'm going to show you a tanglegram. Tanglegrams look kind of like this. It's just 2 phylogenies facing each other. Do not be overwhelmed. In this case, the phylogeny on the left is going to be inferred from nuclear SNPs, and the phylogeny on the right is going to be from the mitochondrial genomes. Because nuclear and mitochondrial genomes have different patterns of inheritance, they can tell us different evolutionary histories. And mismatches or disagreements between those two can be identified by crossing lines between this tanglegram and may be indicative of gene flow. So here's our real data. I'm going to add to this. Nuclear on the left, mitochondrial on the right. The vertical tan and teal bars indicate the Rudolus and Gaperi sorted genotypes respectively. Within each species, I'll add an additional layer of information showing populations. So Rudolus populations here are indicated in shades of red, and Gaperi populations in shades of green. The blue populations here, however, shown mostly in the middle, are discordant or mismatched, which we can better see when I add lines connecting the individuals from the nuclear phylogeny to themselves in the mitochondrial phylogeny. All of these individuals have Gaperi nuclear ancestry, but a Rudolus mitochondrial genome, suggesting a history of introgression. Looking at this geographically, that discordance occurs in the very center of these contact zones in Southeast Alaska, or SEAK, and British Columbia, or BC. Interestingly, introgression is always occurring in the same direction, northern moving into southern, but if we zoom into BC, we can look at these giant teal dots, which are actually pie charts. These pie charts represent nuclear ancestry proportions for voles with mito-nuclear discordance. So all of these voles have a Rudolus or a tan mitochondrial genome, but looking at the giant teal dots, they have almost no nuclear introgression. They're almost completely Gaperi nuclearly. There are, however, small slivers, if you really squint, and the slivers of Rudolus introgression are also larger the closer we get to that contact zone. So we wanted to test if those tiny slivers of introgression, nuclear introgression, are significant. To do that, we performed an ABBA-BABA test such that a positive D statistic would indicate introgression from Rudolus into that population. And we found evidence of significant nuclear introgression from Rudolus into most of these populations, with the highest amount occurring near the center of the zone or further north. So this pattern suggests that this is a moving contact zone with Gaperi expanding north, capturing the Rudolus mitochondria, and then leaving a trail of mitochondrial introgression in its wake. Now, what is occurring in Southeast Alaska just on the other side of the Coast Mountains? Here, again, are pie charts of nuclear ancestry, even though they don't look like pie charts. Again, all of these individuals have a Rudolus mitochondrial genome, but there's a clear break in Rudolus nuclear ancestry compared to Gaperi nuclear ancestry. Again, however, we found significant evidence for nuclear introgression, but less than what was observed in British Columbia. So even though the species do clearly occur in close proximity here, contact may not be as recent in Alaska as it is in British Columbia. And this makes a little more sense if we compare the landscape here. So this is LeConte Bay, which is about a mile wide and connects the ice fields of the Coast Range, the Coast Mountain Range, to the ocean. So this is a serious biogeographic barrier that voles really can't get across. So we believe that Alaska represents this older contact zone, and that we're now seeing the repeated capture of the northern mitochondria by expanding southern populations, first in Alaska and then in British Columbia. To expand on this, we've now sequenced whole nuclear and mitochondrial genomes for each parental species and both hybrid zones, and are testing for selection and biased introgression. That's because introgression was only occurring in one direction. So we wanted to know, is there a selective advantage to the northern mitochondria? And is that tiny sliver of rootless introgression disproportionately associated with mitochondrially associated nuclear genes, so nuclear genes that are involved in mitochondrial function? Mitochondria are functionally important for metabolic processes, of course, such as energy and heat production, but they're also important for ion homeostasis, apoptosis, and immune signaling, among other lesser appreciated aspects. To carry out these processes, mitochondria have their own genome with 13 protein-coding genes, but those 13 genes are not enough on their own, and they must interact with more than 1,000 nuclear genes to carry out all of their functions. First, we wanted to test for selection in the mitochondrial genome, so we calculated a ratio of non-synonymous to synonymous substitutions, or the DNDS ratio. A higher DNDS ratio can indicate positive selection. And we did this for each mitochondrial gene independently, first for Gaperi to Rudolus as a baseline, and that is indicated by the light gray dots shown on the right. We then did this for discordant individuals in Alaska, which are shown here in dark blue. And you'll notice they're almost the same. So this suggests that there is not positive selection occurring in the introgressed Southeast Alaskan mitochondrial genomes. But we did find evidence of potential positive selection in the introgressed mitochondrial genomes in British Columbia. So this could indicate that these mitochondrial capture events are pretty rare, but they may be highly beneficial to the southern species, leading to a selective sweep through the population. But we do still need to test for relaxed purifying selection to fully characterize this system. Next, we wanted to dig into this biased nuclear introgression. So we built a gene tree for all known mitochondrially associated nuclear genes, or NMT genes. This is the tree for the ABCD1 gene, which I don't know the exact function of, but all of the individuals shown in teal are discordant. Everything on this phylogeny has a rudolus mitochondrial genome, but obviously they have different nuclear genomes. Some of our trees look like this, indicating essentially no introgression at that gene. They sort by species, Gaperi and Rudolus. We also measured DXY, a measure of absolute genetic differentiation for each discordant individual in comparison to Rudolus, and found that DXY is high in the absence of introgression. So here it's just over 0.5. Some of our trees look like this, while others look more like this. So some discordant Gaperi individuals group perfectly with Rudolus and have a DXY value that's approaching 0, indicating complete introgression of the Rudolus allele in these individuals, while other discordant individuals have these more intermediate DXY values and fall out on kind of short intermediate branches in the phylogeny, indicating they may be heterozygous for Gaperi and Rudolus alleles. And for others, we see almost complete introgression of the Rudolus allele into Gaperi populations with DXY at essentially 0. Now, we then calculated DXY for every discordant individual at every NMT gene and compared that to all other nuclear genes that are not associated with mitochondrial function and tested if there's a bias of introgression. And we do see a biased introgression of mitochondrially associated nuclear genes, but only in British Columbia. And we're trying to figure out why. So not in Southeast Alaska. We are currently running sliding window analyses of local ancestry across the entire genome to identify these shared blocks of Rudolus. So you can see all the chromosomes on the bottom, all of the individuals in rows, and I've zoomed in on one here, and you can clearly see on the far left this fixed introgressed Rudolus block. And so now we're doing some Go analyses and functional analyses to understand what's introgressing why and associating that with recombination rates, as well as using those block sizes to date some of these hybridization events. So summarizing this last example, we do see repeated capture of the northern mitochondria by the southern species. We see preliminary evidence of positive selection, but only in BC. And we find evidence of biased introgression, but only in BC. So we're in the process of exploring these data to understand what that means. Perhaps the age of these hybrid zones has allowed the signal to change in Southeast Alaska, which is much older, whereas BC is an active mixing zone. So ultimately, understanding the repeatability of hybrid outcomes is super informative for wildlife management, as hybridization in mammals is increasingly caused by introductions, translocations, or even accidental invasions. Overall, we've examined a few different dimensions of change today: taxonomy, geography, and time. And surprise, surprise, we found that species responses are idiosyncratic, like most things in biology. They depend on a combination of geography, environment, and the degree of divergence. Recall that viral diversity across the mammal phylogeny differed from expectations, with greater viral richness found in shrews than in rodents or bats, urging a broader view of pathogen diversity in mammals. We found that traditional ecological niche models fail to capture dimensions of risk that are relevant to spillover, but integration of these models with simulations may better inform public health risk. And that voles are experiencing this repeated unidirectional adaptive introgression of the northern mitochondrial genome into southern populations with almost no nuclear ancestry or no nuclear introgression. Together, these results urge expanded investigations of species responses to change as they're idiosyncratic and context dependent. And the current rate of change challenges the adaptive capacity of species. So next, I plan to bring these different aspects of my research program together to address questions about how hybridization in mammals shapes patterns of pathogen transmission and evolution across space, time, and taxonomy. So there's preliminary evidence in rodent hybrid zones in Europe that have shown that host mixing can lead to viral reassortment, specifically in hantaviruses, and this can lead to the evolution of new and potentially more infectious variants. As such, I have a series of funded projects focused on hantaviruses, avian influenza, chronic wasting disease, as well as the coccidioides fungus, which I look forward to exploring in small mammals. And with that, I'll acknowledge my lab, who I couldn't do any of this without, the global team of collaborators, and my many funding sources, NIH, NSF, USDA, and now Department of Defense. So I'm happy to take any questions.
Let's stand apart from each other. Is there any questions for Jocelyn?
Great talk, thank you. Oh God, it's so loud. So, is there anything in their life history traits that would increase the discordance between the nuclear and mitochondrial genomes?
Not that we're aware of. We've looked at like dispersal distances, like are we expecting some type of sexual selection or sex differences in how far they're moving to find mates, and it looks, we have limited information on this. From what we have, there's nothing that stands out. They're also like almost identical when you look at them. There's lore about one looking more red than the other, but the only way to identify the species is either genetically or looking at the hard palate on a cleaned skull. So just looking at them, I mean, we trick ourselves and tell ourselves it's more red, but they're pretty identical to us. There's got to be something though.
Tim, we'll try the mic to see if there's no feedback here.
Great talk. So as you're going through these biorepository samples to try to find your 1,000 species, are you finding that they have good data on those specimens in terms of like their health and their environment or behavior or anything like that?
No, I wish. No, I think it could definitely be better. We don't have a lot of information. I do think most tissues, I mean, museums started collecting tissues in like the 1970s following advent of PCR, and I think museums evolved rapidly to scale the needs of the scientific community. Historically, like a chunk of muscle was like wrapped in tinfoil and in a freezer, and now like obviously we're using giant liquid nitrogen dewars and 2 mil tubes, and everything has changed. I think think some groups are adding an additional dimension. So this is part of holistic sampling. For part of my postdoc, for example, I was doing physiology work. I've mentioned that to a few folks here. And we've paired that physiological information, so basal metabolic rates, with individual specimens. So those animals were euthanized for transcriptomic and genomic analyses, and the utility of that information should go beyond that one study. So all of the physiological measurements that we took from animals in the field and in the lab are associated with them. I think museums are now battling with how to serve digital data and whether we can serve all of that or we're using repositories and connecting with repositories, that the physiological data is elsewhere, but that catalog number gives that permanent link so that the information can be associated. We need more of that.
Allison.
Great talk, thanks. I was curious about your decision to only screen for viruses when you were, and I was wondering if you had bycatch of other kinds of bacteria or fungi or eukaryotic. Yes.
Yes,
okay.
We do. So we only enriched for viruses, but yes, we sequenced everything. So we definitely have bacterial bycatch and fungal bycatch, and we will be digging into those data sets as well. NSF just liked funding the virus stuff. So that's what we have data on first, because our collaborator Tony Goldberg at the University of Wisconsin and Yuri Wolf and Eugene Koonin at NCBI they had the pipelines established for the viruses, and they're virologists. So yeah, we are doing that. Good question.
Other questions?
Thanks. Really enjoyed the talk. A technical question and then a science question related to it. What tissue type are you using and/or is best for getting these virus samples? And then the broader question is, you've done all this and you have these thousands of viruses. What were there any, I guess, do you have expectations about what, biochemically would be different about something that's carrying a lot of viruses versus species that are carrying few, or predictions about their genomes, what those might look like? Or
Okay.
Thanks.
So first question, what tissues are we screening? So ideally, we are screening lung, liver, and spleen for every individual. So 600 for phase 1 and then 600 in phase 2. One of the major barriers we've run into, Tim, is the availability of all of those tissue types through museums. So some museums adopted holistic collecting right away and sampled multiple tissue types, and others have been slower to adopt change. And we are now trying to accommodate uneven tissue type sampling bioinformatically. So we obviously we're going to get more viruses if we sample more in different tissues. We get more respiratory viruses sampling lung, but we get a lot of viruses in spleen, and liver tends to just be the one that's most available because it's high volume in the host. So we've done all three. And we do extractions in isolation. We pool for sequencing, but we can go back to tissue-specific extractions so we can tell where we're detecting those viruses. So for individuals that are testing positive for something interesting, we want to know where we got it. We can test more tissues, but that was just based on resource availability. The second question, geophysical or biochemical expectations of the host.